I'm Dr. Monty Powell from City of Hope, and this is On the Edge of Breakthrough, Voices of Cancer Research. Each episode, we bring you the minds behind the science, the stories behind the data, and the breakthroughs that could change everything. Let's dive in. Welcome back, everyone. Our title is On the Edge of Breakthrough, of course, and I have to tell you today I'm a little bit on edge because I'm here with the Cancer Center Director of City of Hope, the Chief Scientific Officer, Dr. John Carpten. John, welcome to the program.
Good morning, Monty. It's great to be here.
Oh, well, I've been waiting for this, the opportunity to sit, across the table from you and really, hear about your vision for City of Hope, what you have planned for the future. But before we get to that, I wanted to start sort of with your origin story. It's really a fascinating journey that you've had in terms of oncology research. And it all begins with your upbringing, I would say, in the South, right? And tell us about that.
Yeah, I had an interesting upbringing. My father was in the Marine Corps. I was born in Hawaii. Kaneohe Bay Marine Corps base. Okay. And after I was 2, he was sent to Yuma, Arizona, which is just barren desert, but there's a Marine Corps base there. So I spent a short stint of my childhood in Yuma, probably 5 or 6 years there, sort of, getting into first grade in Arizona. And I think that was when, I really began to express my interest in life science. It was just natural. I was the kid out in the desert trying to catch scorpions and spiders and bringing them home.
Oh my gosh. Okay. I'm sure Mom was thrilled about that, right?
Mom was thrilled about that. And after my father retired, the decision was to move back home, quotes, air quotes, which was the Mississippi Delta. Both my parents were from the Mississippi Delta. So they packed us up and we moved to Leland, Mississippi, small town. It's famous for Jim Henson, who founded the Muppets.
You're kidding. Okay.
Yeah, so there's a creek that runs through my town, Deer Creek, and that was the inspiration for him creating the Muppets.
It's a fun fact.
Yeah, it's a fun fact. And so I may have been 7 years old when we moved to Mississippi. I think I sort of expanded right on my interest in life science there, because now I'm out of the desert and I'm into essentially what was a jungle at one point, right? You've got, we're 4 or 5 miles from the Mississippi River, so it's the richest soil in the world. And so there's a lot of life.
Okay.
Right? And so instead of being in a desert, I'd be in the cotton fields.
Okay.
And, I was the weird kid who thought that caterpillars was the most beautiful organism on the planet, right?
Better than scorpions.
Better than scorpions, man. So yeah, I've always had this interest, this innate, just natural interest in life science. Growing up in Mississippi, the other thing is sports.
Okay.
Requisite.
Right.
Run, throw, catch. Hit didn't matter, right?
Okay.
I just had some natural ability, so I was able to, use that as another way to sort of, become more social, engage, be a part of the community, a part of all the kids in the community growing up. And there's not a lot of opportunity in the Mississippi Delta, right? Small town in the middle of the cotton fields, right? And from a young age, I sort of figured, I want to parlay this into something. Professional, right? Research, right? Medicine, something to that degree. But then there were the sports, right? And I was excelling in sports as well.
And ultimately, this took you to Lane College, is that right?
It took me to Lane College. And I think by the time I got there, I was this weird creature. I was a nerd surrounded by a jock.
Okay.
Right? And they were dueling,
Okay.
constantly dueling. And, I had a great biology, professor, and Samuel Braxter, who's just a fantastic mentor, and really helped me navigate right through college and biology. What do I want to do? How do we tweak and tune your curriculum to get you into med school or graduate school? But again, there was football.
Okay.
And, I was excelling on the football field as well.
No kidding. What position, if I may ask?
By the time I got to college, it was sort of a linebacker safety hybrid.
Okay.
If that makes sense.
Okay. Yeah. Two very different positions, I guess.
Yeah. Last line of defense. If they get by John, it's a touchdown. That was the typical saying. And,
okay.
I played defense.
Awesome. Okay.
Yeah. So, that's a short version.
So, and then, so this interest in the sciences in college, ultimately, how did that sort of take you to Ohio State for your PhD?
I had a term paper I had to write for literature, and I can't remember. I think it was my junior year. And this is when we had libraries, meaning you had to go into the building.
I remember those, yeah.
It's something that I'll never forget because it was such an important moment in my life. I walked into the library and there was a little lobby area where there was a few chairs and a sofa and there was a table. And then they would have a few periodicals just sitting there. And I can't remember I think it was a Life magazine. And the cover was red. That's what caught my eye.
Okay.
And I went over and looked, and it said something like genetic engineering, the wave of the future.
Okay.
Or something, right? And I sat down and I opened it and I started reading through this article. And I said, that's what I want to do.
No kidding.
Like, I swear to God.
Well, and I remember that time frame because back then, genetic engineering, it was sort of a pipe dream, right?
This was 86, maybe?
Yeah.
87?
So it's almost the same way that people talk about flying cars now, right?
Yeah, exactly.
Yeah.
Genetic engineering. And so a few months later, one of our faculty members took a group of us on a graduate school visit to Ohio State. And Ohio State would hold this graduate and professional school fair where they would invite, juniors and seniors from college to expose them, right, to, medical school, law school, graduate school, just to meet professors and meet faculty and meet some of the students and just get an idea of what it's like, right, to be in that environment. And it was interesting. It was a massive room full of kids and individuals from faculty members or administrators from the different colleges and would come by. You'd sort of sign up what you wanted to do. And so I'm sitting there and, we're the law school, bunch of kids get up, we're medical school, we're dental school, and the room is getting smaller and smaller, fewer people, and I'm the only one sitting there. And then this guy, Paul First, walks in. Are you John Carpten, molecular genetics? I was like, yeah, that's me. Right? So there wasn't a van, it was his car, right? I was the only one who signed up for the Department of Molecular Genetics. And he drove me over to the building and, I met the chair and some of the faculty members, and they talked about the research that they're doing. Roy Tassova, who was studying wound healing in lizards and amphibians, and Phil Perlman, who was studying the genetics of yeast. I mean, it was just amazing, right? And I said, this is what I want to do. And I remember having to go back, and a bunch of my teammates were going to New Orleans for tryouts for a couple of Canadian League football teams. I had, run for some scouts and, but when I got back, I said, that's what I want to do with my life. I'm going to Ohio State. Called my mom. We had a conversation. That was it. That's how it happened.
That's amazing. You
packed up my car and drove to Columbus.
Amazing. But you may be surprised to hear this, but I've never had that conflict between football and science, just, given my build, but it's just never happened.
Hilarious. Yeah.
But tell us about your PhD work. What were you focused on during the PhD?
Yeah, it's interesting. I guess I'm a little embarrassed, but, it's my story. I had never had any experience in real laboratory research. I'd done some work in high school. So the U.S. Department of Agriculture has an experiment station outside of my hometown. It's a massive research facility where they study things like the migration pattern of the cotton bollworm moth, right? I mean, things of that nature, right? But I actually had summer jobs out there and had projects that took me to the state science fair. But it wasn't molecular research, right? In college, it was dissecting a frog or a rat or things of that nature. I didn't have the experience. Some of the kids who were in my graduate school class, had done internships during the summer at universities and actually knew what a pipette was, right? So when I got there, I had no idea what I was getting myself into. So I was a little behind, and I found myself on academic probation. My advisor basically said, we're going to put you on a 500 level biochemistry class, right? I said, okay. So I took that step, and it was funny. It was a week. I said, oh my God, it's just biochemistry. DNA is a chemical.
Yeah.
It's a biochemical. And it just clicked.
Okay.
Right. And so I began to think about it from the perspective of chemistry. And then there was another epiphany where, you know, I began to approach it from the perspective of, if I close my eyes, can I imagine what's going on inside of the tube, right? And these were my sort of approaches to help me better understand not just what was happening didactically, but what was happening practically in the laboratory. And it was at that moment that I put the two things together. After getting out of that 500 level class, it just exploded. I was the second person in my group to defend, right? Everything just clicked. And so I joined the laboratory of someone who was fresh out of their postdoc, Arthur Burgess. He had just finished his postdoc. This was his first faculty position. I was his second graduate student. And he had worked in one of the laboratories that helped identify the gene that caused muscular dystrophy, the Duchenne's muscular dystrophy. And his goal was to be a part of the team that identified the gene that caused another rare pediatric disorder, spinal muscular atrophy, which was another type of muscular dystrophy, muscle wasting, innervation of the muscles. Again, the kids get these diseases, and it's just horrible. And so Arthur began his faculty and his professional career at Ohio State, and I was one of the students that joined his lab. And subsequent to that, during one of the Molecular Genetics 701 courses, there was a presentation on a new methodology for human genetics called yeast artificial chromosomes, where you could take large pieces of human DNA and clone them into a vector and transfect that vector into yeast cells and then propagate that large piece of human DNA. And so you can imagine you can generate a library of these clones that would essentially represent all of the DNA across the entire genome. And then if you could sort of amplify the ends of that, you could generate PCR products and find contiguous pieces of DNA that would represent a long stretch of human DNA. And so Arthur's lab had identified a region on human chromosome 5 that likely contained the gene that would cause that disease. And so my role in the lab was to build out the physical map of that region of human chromosome 5 that would allow us to map new genes to the region and help us identify the mutations in the families that cause that disease. And so this was essentially the beginning of the genetic revolution. So Francis Collins and Mary Claire King, they were trying to identify the breast cancer genes at that time. Cystic fibrosis had just been cloned, right? So it was just great timing. I got in the right field at the right time. And here we are.
Yeah, incredible. And it's amazing that you can pinpoint this all to looking at that journal cover, right, in the library.
At that journal cover in the library. It just was I looked over, sat down, opened it up. That's what I want to do.
Just a real turning point. You can see sort of a through line to, all these other major accomplishments.
That was it.
You'd mentioned Francis Collins, and I know that he's been a mentor to you of sorts over the years. I know that your journey took you from Ohio State to the NIH, right, for postdoctoral fellowship. So tell us about that continuum and how that came about.
Yeah, it's interesting. Again, genetic revolution. Francis Collins and Jeff Trent had worked together at the University of Michigan, and the National Institutes of Health was looking for new leadership for, at the time, what was the National Center for Human Genome Research. And Francis accepted the job. So Jeff and Francis became the director and scientific director for the National Center for Human Genome Research. And I was finishing up my dissertation. It was about, maybe little under a year out from defending and thinking about the next steps. And Arthur and I had been talking about things, on one hand, it was industry, go make some money, get married and, have a regular life, or do this academic thing, right? And Arthur knew that I was a huge fan of Francis. Like, Francis was my hero, right? I had heroes in football and I had heroes in science, and Francis was probably my biggest hero. Ohio State's cancer center at the time was really small. There might have been 5 or 6 people doing cancer research at Ohio State at that time. And Arthur encouraged me to send my CV into the Genome Center. He said, you love Francis, why don't you at least send your CV and see what happens? And one day the phone rang in Arthur's office and he said, hey, it's the Genome Center. They want to talk to you. And it was Jeff Trent.
Jeff Trent. And just for our audience, Jeff Trent presides over TGen, a City of Hope affiliate. And, I have to tell you that Jeff is one of the most accomplished and also understated guys in the field of genetics research. Is that a fair statement?
Absolutely. And he's a wonderful human being.
I've got to agree with you there. Yeah.
And so he gave me a call and he said, hey, see, you've done positional cloning and, physical mapping. We're going to do a lot of that work here at the Genome Center. You've got all these skills. We'd love to invite you out, and talk about a postdoc position. Wow. So I jumped on a plane, flew to Bethesda. And of course, I wanted to work for Francis, right? Francis was leading a series of national and international studies to identify genes linked to diabetes. And one of the studies was in Africa, and another study was in Finland. He was working with Mike Binky at the University of Michigan. And so the agenda was to meet with all of the various faculty members. It was a veritable who's who in genome research at the time. And I remember the moment I went to meet with Francis. He and Larry Brody was one of his postdocs. The gene for the BRCA1 gene had just been published.
No kidding.
Like a week or two before my interview.
Okay.
And I walked in the conference room and Larry and Francis were holding autoradiographs up to the light, looking at the mutations in their BRCA families. When I walked in when I opened the door, I walked in and they're like this. Oh, you're John Carpten. Yeah, have a seat. I'll be with you in a few minutes. Like one of those moments, right? This is Francis looking at SSCP gels, right, in the light. And so we sat down and he said, well, John, I reviewed your CV. It looks great. You have all of the appropriate skills, but I just don't have room. So I have 5 postdocs already. And, as a postdoc, you need your own thing. You have to have a project of your own. That's really the only way you'll be successful. And I just don't have room. So of course I was disappointed. So I circled back with Jeff at the end of the day. Jeff was my official host. He said, how'd it go with Francis? I said, well, he doesn't have room. I'd already started thinking about next steps, right? Of
course, yeah.
And he said, great, I have a postdoc for you.
Oh, you're kidding.
He said, they just cloned the breast cancer gene. He said, I'm working with some investigators at Johns Hopkins to identify the gene that might cause hereditary prostate cancer. Okay, prostate cancer. What's that, right?
Of course.
I had no real background or understanding of cancer. It just wasn't something that I had really focused any of my attention on. And I thought about it, and we sat there and talked for about 30 minutes and said, this actually sounds pretty cool, right? Especially all of the press that the breast cancer gene got.
Around what time frame was this?
94.
94, okay.
Yeah, I called Jeff back and said, sign me up, right? What are the next steps? So I joined Jeff's lab. We moved to Maryland, I believe in October.
Wow.
And that's when it all began. And we created a group called the Prostate Investigation Group, or the PIGS.
Okay.
And it's funny because Jeff got flack, I think, from some of the NIH leadership. We hear that there's some group, you can't use that term as an official. Like, come on, man. It's just, it was a fun thing for us to do.
I know for the clinical trial space, the NCI, they don't love acronyms. That's the one thing I've heard about them.
They did not like that acronym, but we rolled with it. And, Jeff was great. And so it was me, and there was another postdoc, Jeff Smith. And we began a lot of work genotyping DNA samples from families that had multiple men who had been diagnosed with prostate cancer at a relatively early age, sort of the indicators that the cancer might be inherited. So first degree relatives, a father and multiple sons, or a grandfather, then the father, then the sons, right? So first degree relatives, multiple first degree relatives diagnosed with prostate cancer, collected blood samples. The blood samples, that work was done at Hopkins. Samples were sent to us, to our lab, and we did all the genotyping in the laboratory. And so Jeff Smith and I had this lab in Building 9, which was one of the older buildings on campus, and we had these massive speakers, and we were like the only lab in the building, and we just turned the music up and eat Pop-Tarts and drink espresso.
Okay.
And
Pop-Tarts and espresso, huh?
Man,
Okay.
yeah, that's how you get 14 to 18 hours, like, every day for multiple years. And we just did it. And before we knew it, there was a science paper. And
tell us about that paper, if you don't mind.
yeah, it was the first paper that reported on a region of the genome that likely harbored a gene that would be associated with hereditary prostate cancer. So it was that first step to get to the actual gene and the mutations, right?
And I just hope our audience appreciates how challenging this type of work was at the time, right? I mean,
there was no
these
genome.
days,
There was no
right?
human genome.
Yeah, I mean, these days you pop something into a sequencer and boom, you've got your answer in a couple of minutes. Then it was a lot of really heavy-duty manual labor, if I may say, right?
Yeah, you had to piece it together.
Okay.
It was like a puzzle, building a puzzle. Now, the cool thing was, of course, Francis was also leading the Human Genome Project. And so as we mapped out this region of the genome, my laboratory led the efforts to build out the yeast artificial chromosome map, which I had done as a graduate student.
Right, so this kind of leans on that experience.
Exactly. And then there were higher resolution clones, right, to build out a more detailed map. And so we did that work and then mapped the genes for the region. Now, the cool offshoot of that was that we built out this 25 megabases of human genome real estate. And so we were able to engage with the Human Genome Project to say, we've mapped out this region of the genome. Why don't you use our clones as part of the Human Genome Project for that region of the genome? So I began to become more involved in the actual Human Genome Project and was one of the human chromosome 1 leaders. So there were different teams working on different human chromosomes. And Jeff really gave me the freedom to operate, right, as a postdoc, to build the lab, build the team, lead the projects, right? He oversaw it, he resourced it. He would join our lab meetings, but he allowed me to take the lead on everything. And so it, really opened a lot of doors for me, Monty.
That's so Jeff, to kind of preside over things, but in a very sort of non-dominating way.
Exactly.
Right?
Exactly.
Just to give you a lot of creative direction.
The creative direction and the opportunities to innovate and think outside of the box. And I believe it was 97 when I had another epiphany in the lab with Jeff Smith. And, we were standard, espresso Pop-Tart day.
And espresso, right.
Right. Bob Marley blasting on the speakers. And I was sitting at my desk in front of my computer and we were analyzing genotype data. And I had this, I don't know, it's like a bubble, right? I wonder how many of these families are African American.
Okay.
Right? I mean, it was just a question that popped into my head. Looked over to Jeff Smith. Hey, Jeff, do you know what the racial background of the families are? He said, no, never even thought about it, John. I said, I wonder if they have that information. He said, reach out to Bill and Sally at Hopkins. And I did. They said, we had about 100 families that they had recruited into the study. I'm thinking, I don't know if you've ever visited Johns Hopkins, but where it resides in the city of Baltimore.
Sure. Yeah.
I'm thinking it's got to be a substantive percentage or a proportion of the families. And it was like 2 of them.
Okay.
And I'm like, wow. And so, from that moment, it was like, how can I create a study similar to this, but focused on African Americans, given the fact that Black men are 2.5 times more likely to be diagnosed with prostate cancer, maybe 2 or 3 times more likely to die from the disease. Shouldn't we have a study, right, that sort of helps us understand what the potential role of biology and genetics might be?
So I'm just registering this now. The study, the 100 families that you'd had, only 2.
Only 2.
That's so interesting. From Hopkins. I mean, that's
Hopkins.
Bizarre. And it really speaks to the issue that we have even today, right?
Even today,
in
Monty.
clinical trial accrual and enrollment and so forth. But carry on.
And so I went to Jeff and Francis with an idea and a concept. I said, I'd like to recapitulate this study, but focused on African American families. So I need to recruit families I want to create a coordinating center to help me do that, pull that off. All of the genetic research would be done here. And Francis said, John, this is a great idea. And Jeff said, I concur. What do you need? Francis came to me and said, hey, there's a scientist or researcher at Howard University, which is a historically Black college in the DC area, Georgia Dunstan, and she wants to do something similar with breast cancer. And so one day Georgia knocked on the door in the lab and we talked about it. And she said, oh, I know some urologists that see, large numbers of African-American men in their clinic. And they're all part of this, the National Medical Association. Monty, I don't know if you know the AMA.
I do, yeah, indeed, yeah.
So they have a urological section. So
Okay.
Isaac Powell was the chair.
Okay.
He was a urologist, African-American urologist who was in Detroit at Carmanos.
Okay.
And so Francis and I reached out to Ike, and he said, I'll pull the entire group together. They flew to Dulles Airport. We drove Francis's old minivan from the campus to Dulles, and we had a meeting room in Dulles.
Okay.
And we met with them, and we laid out our project plan, and they endorsed it and said, we're in to help. We can get you 100 families in no time.
I did a little bit of homework ahead of this podcast, and to my knowledge, I can't think of any other setting in which, there was a sort of focused exploration within an ethnic group. Is that fair to say? Within oncology, I think this was really the first example that I could come up with of looking at, hereditary patterns, genetically based within oncology. Is that fair to say?
That's fair to say. I would say among the first. If there was another similar study, I would probably say Fumiola Patti at the University of Chicago.
Oh, sure. Okay.
Was beginning her work in Africa. And, Monty, it was bigger than that for me. It was about the community.
Yeah.
And so how could we also ensure that, African Americans were involved in the study design, recruiting the family, the nurses, the physicians. The coordinating center was at Howard University. The study coordinator was Rick Kittles. I hired Rick.
Gosh,
we hired Rick right out of graduate school to become the study. Yes.
I got to know Rick well during his time at City of Hope.
Of course.
Yeah.
So Rick became a part of the team. And Agnes Bafubani, who had trained as a statistical geneticist with Joan Bailey Wilson at NHGRI, so it was a African American led study team to focus on a disease that was significantly impacting our community. And it's to this moment, right, one of those achievements, where you just say, okay, I've impacted my community, right, in a very, very positive way. And there's still individuals being trained by me or Rick and some of the urologists who participated. So even though the study is completed, right, it lives on.
Yeah, indeed. I've read this story. It's so different hearing it. And I will say it explains so much of our focus. City of Hope expands into Chicago and Atlanta and Phoenix. I mean, one of the many awesome elements of this expansion is that we can actually capture diverse populations in our research and really try to address this problem.
Absolutely. Everyone deserves access, right, to innovative care.
Yeah.
And that's one of the things I feel that's a responsibility of mine. So, it's not just about the job. It's not just about sort of the work. It's responsibility and purpose. That's what gets me up every day.
Awesome. Yeah, we've only gone through a part of your journey so far. So you did your postdoc at the NIH. I think it's really curious, sort of the path that emerged from this. I mean, this illustrious experience, right? Founding this group, working with Francis Collins, doing this very foundational work. Where did this take you next?
So Jeff and I had some conversations about my next steps, and I had to compete for an open tenure track slot. So there were a couple of different opportunities, right? One was Ohio State.
Okay.
But Jeff and Francis both wanted me to stay at Genome, but the decision among the faculty was we're not going to give him a faculty slot. He's got to compete for it. I competed for it, and I earned that faculty slot. So I joined the cancer genetics branch, and as a faculty member, I think this was 99, maybe 2000. I played baseball in college too, high school and college. And little did I know, when I interviewed with Jeff You know, he was sitting there and he's looking at my CV. I'm waiting for him to ask me a question about science. He said, I see you played baseball in college, right? Little did I know he was trying to recruit me to his softball team.
Are you telling me Jeff Trent plays softball?
Jeff is uber competitive. He formed this softball team and we played in the Navy League and the Army League. You know, business people play golf and do business.
Yeah.
Well, Jeff, we play softball and we talk science, talk shop in the dugout or before the game, throwing the ball around. So one day, I think it was in 2001, he approaches me, said, hey, you know, I really want to apply the genome to human health. I just can't figure out how to get it done here.
Okay.
He says, so I'm thinking about leaving. Right. And I'm like, what?
This is during a softball game.
Yeah.
Okay.
Yeah. Things are going really, really well.
Right.
Been talking to some people in the state of Arizona about creating a new research institute. Arizona. I'm like, no way. I'm not moving to Arizona. And one day I was in my office and said, hey, I've got this group of people from Arizona coming and they're going to be in a conference room in a few hours. I need you to, you know, come in, you know, and I have a baseball hat on and, you know, it's just regular day. So I go into the conference room and contingency of individuals from the state of Arizona, the mayor of Phoenix and Bennett Dorrance and some of the major benefactors.
Okay.
And so Jeff introduces us and asks us to give a little spiel on what we were working on in the lab. And there was no formal presentation. Jeff did a presentation, but we all just introduced ourselves and went back to my office and didn't think much of it. And a few months later, you know, Jeff said, hey, I'm going to take this position in Arizona. I'd love for you to come with me.
Okay.
My wife's like, dude, I'm not moving to Arizona. You get that out of your head.
Okay.
And so it was a really, really tough decision. And I think it just boiled down to the opportunity for a new build.
Okay.
So when we got there was no building.
Wow.
There was no foundation. There was a empty lot in downtown Phoenix. Then before we knew it, a couple of years later, there was the TGen building and we moved in.
So you get out there to TGen and what's it like, you know, just starting this practice first in a Quest laboratory, then in this, you know, beautiful new facility. What were some of the major accomplishments in those first couple of years out?
Jeff is a very entrepreneurial scientist. And, you know, he respects the academic mission, but understands that to drive things into the clinic, to really impact patients, right, it requires the development of products, right?
Oh, yeah, indeed.
Right? And so, you know, it really allowed me to begin to think differently about science and how we do science and what the outputs of our science should be geared towards.
And with all due respect, that's a very different focus from the NIH, right? I mean, it's
very different focus.
Yeah.
And this is, I think, one of the reasons why he wanted to sort of move on. And then there was Dan. So Dan Von Hoff.
Dan Von Hoff. Okay, great. He's another big part of this story, I think, right?
Yeah. So that partnership was just something I had not really understood or experienced, where you have Jeff with the technology, the innovation, the entrepreneurial spirit, and then you've got Dan with this missionary zeal to help patients and save lives, especially for a tough disease like pancreatic cancer, right?
Indeed.
And so this translational platform that we were building, it wasn't about research. It was translational research. And I believe TGen was the first organization to use the word translational in its name.
Okay. Yeah, I didn't dig that up, but that's probably absolutely true. That's interesting.
If someone shows me something different, I'll take a look at it. But I believe we were the first to use translational in the name of the organization. And so the two of them, and it probably within the first 6 months, I just got it, right, Monty? It was really about, you know, how do we innovate the technology? How do we accelerate and advance the research? How do we ensure that the research that we're doing is focused on outputs that have the potential to impact the lives of patients?
I love that. You know, just for the audience who may not know Dan Von Hoff, maybe I'll just give him a quick spiel. You know, Dan has just had this transformative impact in oncology, and he remains very much involved in sort of paving a path for now application, I think, of precision medicine, right? Yeah.
And this was the foundation of precision medicine. How do we utilize genomics, right, to improve the practice of medicine? And that was the focus of our work at TGen, right? What are the discoveries that we can make, again, where the results, right, could impact the practice of medicine and improve the lives of cancer patients? Jeff and Jeff's collaborator, Michael Bittner, had created one of the first microarray technologies when we were in Bethesda.
No kidding. Okay.
So they were printing genes on glass slides.
Okay.
Pat Brown and David Botstein were doing the photolithographic printing of oligos,
Wow.
which was the Affymetrix, became the Affymetrix
Oh, I remember the
technology.
Affy chip. Yeah.
Jeff and Michael's became Agilent's
Okay.
technology. And when we moved to TGen, Dan had the idea, why don't we clinically validate this assay, and then
Okay.
we'll test the tumors, gene expression profiles, and then we'll make the treatment decisions based on the profile of the tumor.
Got
So
it.
this was the beginning and foundation of precision medicine.
Wow.
David was really focused on data science and bioinformatics. On the other side of the hall was my group, the Cancer Genomics program. And we were using different sequencing technologies and different approaches, but trying to ask similar questions. It may have been 2008 or 9. I had a knock on my door, and it was David and one of his colleagues. And they said, hey, we see you're using this ABI solid next generation sequencer. We'd love to, grab some of your data to develop tools to identify variants and mutations. I said, awesome, right?
Yeah.
So we sat down, and they were using Illumina sequencers. I was using ABI sequencers. And we were seeing and observing what Dan and Mike and those guys were doing. We said, we should be doing this with sequencing. And so that was the foundation of creating a next generation sequencing based clinical laboratory. And so David and I set out, right, to build a lab based on next generation sequencing. And I remember going to Bennett Dorrance, who's one of the major benefactors of TGen, who's a philanthropist, with the idea. And I remember going to his foundation and presenting the idea. And they funded me, I think, $1 million a year for 5 years. And I built a lab, and we called it Ashion Analytics.
Okay.
And that was the clinical laboratory that was developed by David and I at TGen.
You know, I have to tell you, in my clinical practice, I was one of the highest end users of the Ashion platform. I loved it. You know, a whole exome, RNA-seq. I thought it really, did a lot of good for my patients. Around this same time, I wanted to call attention to a paper that I actually think has really had a transformative impact. This is a Nature paper that you'd first authored, identifying AKT, right? Which, I want to underscore for the audience what a dramatic impact this has had. To this day, right, we have clinical trials targeting AKT, different elements along the, what we call the PI3K AKT signaling axis. And, I think your work was really foundational there, right?
Yeah, that was probably 2005, I think. We began collaborations with Eli Lilly. One of the things that Lilly wanted to do was to identify new targets for therapy for cancer. And so we devised a study where, and there were two major components. One was a large molecular profiling study that I led with Mike Bittner, and the other was more of a functional genomics study that was led by Spiro Musses, one of the other faculty members at TGen, where he was using siRNA to knock out genes and create perturbations in cells, and then see how those cells would, the behavior and biology of those cells would change under different pharmacological conditions, right? So that there was that functional genomics. And then we performed, which to some degree was sort of a pre-TCGA type study, where Eli Lilly acquired about 600 tumor samples. And the question was, let's profile and sequence and perform microarray analysis on these tumors to identify novel therapeutic targets. They wanted to focus on kinases. And so my laboratory created PCR primers to PCR up the genes.
Okay.
And then we would PCR the DNA from the tumor samples, right? And then we would perform the analysis and look for new mutations. And I remember during the design of the study, we were looking at some of the major cancer oncogenes, and including the AKT family.
Yeah.
And there are two major regions in AKT. One is the plextrin homology domain. The other is the kinase domain. Lilly really wanted us to focus on the kinase domain. And I said, no, I want to do the whole plextrin homology. I want to do the whole thing.
Okay.
And lo and behold, right, this mutation, E17K popped up. And Jim Thomas at Lilly was super, super excited about it. And so we went ahead and did all the biochemistry and showed that it was an activating mutation that credentialed AKT as a bona fide oncogene in human cancers. And so it was really, really amazing work. And that was published in Nature.
Incredible.
And I think there was one other paper, though, Monty, that I don't know if it's gotten as much publicity, but I also see it as one of the major accomplishments. And that was the study with Mitesh Borad and Alan Bryce.
Oh, okay. Alan Bryce, who now heads City of Hope Phoenix.
Exactly, where we identified mutations in the EGFR pathway and oncogenic fusions in FGFR2 in cholangiocarcinoma.
Oh, gosh. Oh,
that
wow.
had never been discovered. And those mutations are now changing the practice of medicine for how cholangios are being treated.
You know, it's funny, I'm a big fan of this literature because I treat bladder cancer as well, and FGFR3 as opposed to 2, you know, plays more of a pathogenic role there, but there's some cross-reactivity between these drugs, right?
Absolutely.
So I knew about AKT, did not know about your work on FGFR2, which is also, I mean, transformative when it comes to oncology care.
Yeah, so Arul Shanayan at Michigan and our group sort of came out with those discoveries around the same time, where we identified oncogenic fusion. So FGFR2 was fused, right, to other partner fusion genes, and they were oncogenic fusions. But the difference was we actually treated patients, right?
Yeah,
and showed that the drugs were efficacious against those tumors that harbored those oncogenic fusions. Really, really exciting work. I believe it was published in PLOS Genetics 2014, 15. But it's interesting going to oncology meetings and going to the cholangio or the hepatic sessions and hearing the oncologists talk about the utilization of FGFR2 inhibitors, EGFR inhibitors, EGFR pathway inhibitors to treat patients with cholangio. And they're trying to move it up closer to first line. So it's really exciting to say, wow, we were a part of that.
That's incredible. And it requires a lot of patience, I would imagine, right? Because you made these discoveries 2 decades ago, and it's really only now, right, that we're starting to see this clinical impact through phase 3 trials, or in the case of cholangio, some of these pivotal phase 2 trials, right?
Yeah, so it was just the application of genomics and again, just that, when we think about precision oncology, precision medicine, those were the beginnings, then the foundation, right, of what we now, call precision medicine, precision oncology. And we kind of served as the precision medicine engine for a lot of groups.
Oh, no kidding. So I have to tell you, it seems like you're on a roll at TGen. Frankly, you're on a roll at the NIH too.
Things are going well.
But what was it that sort of pulled you over to the USC role?
I think there were probably 2 main pulls, Monty. I think one was the opportunity to be in an environment where there was a hospital with patients and the opportunity to sort of provide that innovative care. I think 2 was the relationship with what was LA County Hospital, which serves 70-80% underserved patients. Patients from underserved communities. And again, for me, responsibility, purpose, ensuring that everyone gets access to this kind of innovation.
Yeah, and not to fast forward through the USC experience, but you were doing great at TGen, doing great at USC, and then of course, we at City of Hope come and try to entice you to come and run our cancer center. Tell us about that.
Yeah, I think a couple of things. Mike Caligiuri was one of my mentors as well, and someone who, I've always looked up to, has always been there to, plant those pearls. When Mike moved here, he and I connected and we talked about it. He knew I had aspirations of one day becoming a cancer center director. City of Hope was doing something very different, and everyone was kind of hearing about it and seeing it and like, wow, they're going national. That's pretty cool. And then 3, the relationship with TGen,
Mm-hmm.
right? Sort of, full circle, right?
It does seem like a trifecta in some ways, right? I mean, gosh, the
Absolutely.
ability to like pull in your old partners from TGen to run the cancer center to really capitalize on this, sort of
National
nationalized
model.
vision. Exactly. Yeah, indeed. Indeed.
So again, I think when I look back on my career, Monty, I've been a builder.
Yeah.
I joined a lab in grad school that didn't exist. So someone fresh out of their postdoc building their first lab. And we literally built the lab. Like we put in cabinets, we put in benches, we built it from the ground up. And then moving to the Genome Center at NIH for Jeff and Francis just showing up. And we built that center, which was later advanced to an institute, right? And then Jeff going to Arizona, nothing existed. And we built something from the ground up. Then USC recruits me and says, we want to build a department in the medical school. And I went to USC and built a department that did not exist.
Mm-hmm.
So every stage has been a build stage. And then City of Hope, which provides a very different challenge because it's an exceptional organization already. I mean, it's unbelievable. You're exceptional. You're, top 5, top 10. I mean, right?
You're right.
So this isn't a new build, but it is a new build, right? I think the phrase has been said, we're an amazing established organization trying to become a startup.
I like that.
Right?
Yeah.
And so it's a different kind of build now.
I see so many applications of the skills that you bring to the table, right? I mean, certainly I think at Duarte we've got things humming, right?
Yeah, for sure.
We have a beautiful new hospital that just opened recently in Irvine. And of course, these new facilities that are emerging in Chicago and Atlanta and Phoenix, with really, I would say, world's authorities in respective areas of oncology at each one of those centers. I think that, you're really going to help us, if I'm not forecasting too much here, in getting some of our research out to those areas. Is that fair, just based on previous experience?
That is definitely fair. I think, this offers a really, really unique opportunity that very few cancer center directors, right, or research leaders get to be a part of. And I think, on multiple levels, when you go to Atlanta, right, and you're in Noonan and you're in South Georgia, right?
Mm-hmm.
And you think about that population, the demographics.
Yeah.
You go to Zion, Illinois, right? And you think about that population and those demographics. In Orange County, right? And of course here in Duarte and across our entire Southern California network that we can't forget about. Right, the on-licensed sites, right, all of our clinical partners, Antelope Valley, Upland, right, just so many opportunities to get innovative care out to the masses, right?
Absolutely.
And so that to me is just such a unique opportunity. I feel so lucky to be a part of it.
Oh, that's awesome. We've talked about, this national model that we have. Really at this grand scale, you've been shaping the direction of oncology for a long time, but now very formally through the National Cancer Advisory Board. Can you tell us about that role? It's a huge honor, right, to join that very, sort of selected group.
Yeah, it's been an unbelievable experience, the appointment from President Biden, and not just the appointment, but to chair.
Yeah.
When someone says he wants you to chair, then you're thinking he, right? It's like, okay, it's
incredible.
The President of the United States.
And I imagine
it's funny, I missed the phone call the first time.
Did you really? Oh, God. Tell us that story, if you don't mind.
It's a bizarre story, but I missed it. I saw the 202 and it said White House. I'm like, this is definitely a scam, right?
You're kidding. It said White House on your phone. Okay.
White House.
Okay. But I would think of that as a scam call. Is that fair?
That is fair.
Okay.
And then I listened to the message and I said, this sounds like it could be credible.
Okay.
Right. And then Francis called me and said, hey, did you get this call? And I was like, he said, you need to respond to this call. So I called him back and and that was that. But it's been a great experience, to work with the directors of the National Cancer Institute and, to, work with them on the agenda for cancer research, to help advocate for funding for biomedical research and to make sure that, the Institute is is providing the type of impact that we know it's supposed to provide.
Yeah.
And to be, a part of that discussion.
Well, you have such a massive role here at City of Hope, but it seems to me as though you're still maintaining a very sort of active research agenda with colleagues like David Craig, who you'd mentioned previously, and others. Any particular bits of work you'd like to spotlight from your current research portfolio?
Well, yeah, it's interesting, Monty. I'm a scientist, right? That's who I am.
Indeed.
We had a paper that came out in Nature Genetics a month or two ago where we profiled 500 triple-negative breast tumors that were derived from African-American women. It's one of the largest genomic studies of African-Americans, and sort of detailed the molecular landscape of those tumors, what's similar or different compared to women from other racial and ethnic backgrounds. So really proud of that work and excited about, the future prospects of those results. One of the things that we focus on a lot in my laboratory is around spatial biology. So trying to really understand the tumor and immune microenvironment changes that occur in tumors with a focus on women's cancers. So triple negative breast cancer, ovarian cancer, and endometrial cancer.
Okay.
So we have spatial biology projects running in all three of those cancer types.
Brilliant. And this is really, am I understanding this correctly, to sort of understand the tumor microenvironment where immune cells sit in relation to cancer? Yeah.
Right. And fibroblasts and the interactions of these cells, the various cellular compartments. For instance, one of the studies we're running in ovarian cancer, we're looking at ovarian cancers that were derived from patients who were treated with immunotherapy. Some patients who responded and some who didn't respond, and looking at the differences in the immune compartments, right, in the responders versus non-responders, using spatial transcriptomic approaches and spatial protein approaches. And we're really excited about that new data we just generated. Just really trying to understand the differences that we see in the tumor and immune microenvironment where we're comparing across groups. It may be groups who have been treated with therapy, who responded or didn't respond. In endometrial cancer, one of the things we know is that Black women tend to present with more aggressive disease and have poorer outcomes. So we're building an endometrial cancer cohort where we're collecting tumors from women across racial and ethnic groups and profiling their tumor and immune microenvironments to see if we can glean any clues or extract any new knowledge that might help us better understand what might be driving, right, that more aggressive cancer in Black women.
Yeah, that makes a lot of sense. We have a phenomenal force in gynecologic oncology here, right, who I'm sure is serving as a great partner.
Collaborating with Lorna Rodriguez and some of the other folks on our gynoc team. Really excited about the prospects.
Oh, gosh. We always wrap up these podcasts by asking maybe what I think is the toughest question of all. Our title is On the Edge of Breakthrough, Voices of Cancer Research. What does On the Edge of Breakthrough mean to you, Dr. Carpten?
How are we going to incorporate technology into every aspect of our organization?
I love that. So really bringing all these discoveries in the lab, sort of to the patient.
And how are we going to implement AI and machine learning to help accelerate discovery, right? Help get it into the clinic faster. What is our technology play? Really excited about our new leader for our enterprise technology group, Simon Nazarian, working closely with he and Marcel and some of the folks on our research team, Russ Rockney, Dave Craig, Vitahe, and just really trying to figure out how do we begin to incorporate technology right into everything that we do, because technology is going to change the way we do research, the way we do clinical care, the way we engage with patients, and we want to be at the bleeding edge of that transition.
I love that, and that's so appropriate for everything that we're seeing emerging from our labs right now, from our AI group. I think there's an excellent opportunity for us to really be leaders in that space.
As I look back on my career, there was bioinformatics, and it was how do we genotype, how do we 3 billion bases of the human genome, and you're looking at thousands of all of these data points across all of these individuals. How do we manage that data, store that data, and analyze that data? working with David, all of the work that they did in developing methods and tools and all of the, working in R and working in Python, developing new informatics tools to now where, I think one of the developers of AI was asked a question, as we think about, technology and coding, what computer language should people be focusing on? He said the English language, because ultimately, how do you ask AI a question?
Brilliant. With your own words.
You don't have to pull down some fancy tool off GitHub anymore. You just go to AI and say, hey, here's a list of genes. What does it mean?
Yeah.
Right. And so how can we do that, right, in a very efficient and effective way? How do we train our students and our fellows, right, on how to use this technology, again, to accelerate discovery?
I love that. Gosh, brilliant. I've talked your ear off here. We've gone probably way over time, but John, thank you so much for sitting down with me. Amazing, amazing history. I can't believe this all started by looking at the cover of a life sciences journal, back there in that college library.
Yeah.
It's an incredible story.
Right moment, right time. I can't take any credit for it.
We've promised me you'll do this again. We've got to come back here in a year or two.
Sure. We'll love to, Monty. This is fantastic. Thanks for the opportunity to share.
You got it. Thanks for tuning in to On the Edge of Breakthrough. See you next time for more insights from the front lines of cancer research and care.